Welcome to day 2 of our ADRC meeting. We are ready to kick off our second and final day of sessions here in DC and on our virtual Hubelo platform, all focused around our theme, Progress Towards a New Therapeutic Era for Alzheimer's Disease and Related Dementias. A couple of announcements before we get started. Today's lineup of events includes the ACTC session that will be immediately following our NACC session, kicking things off. Later today, we have a few networking breaks, the clinical core session, imaging core session, and biomarker core session. Lots of great talks lined up for those. Make sure to stick around. But without further ado, I'd like to welcome to the stage Dr. Sarah Biber from NACC. She is our program director, and she's starting off with a presentation on NACC modernization and the new NACC data platform. Welcome, Sarah.
Hello, it's good to see you all again today. Thanks for being here. I'm honored to be kicking off the NACC session today on behalf of the NACC team, and I couldn't be more excited about the other speakers that will be joining for the NACC session today. But before I get started with my talk, I wanted to mention that everything that I'm going to, here are the speakers. I wanted to mention that everything that I am going to be presenting on today was made possible by the incredible team at NACC. I want to thank the NACC team for teaming up to accomplish this important work together. Can all of the NACC team members actually who are here please stand up? And folks can take a look and see who is here. I encourage all of you, all of you to talk to them. Encourage all of you to talk to them about the incredible work that they are doing. Many of them are in the hallway ready to do demos with you on all sorts of new cool stuff that we have going on, so make sure to take advantage of that while you are here. I also want to give a special shout out to the team members who were instrumental in organizing this conference. Thank you, Hannah, Brittany, Eileen, Faith, Jane, Val, and Amy. These conferences are a huge lift, and you guys have all done an amazing job. I want to give a special shout out to Hannah, who has spearheaded so many aspects of this conference and done an amazing job. So thank you guys so much for all your hard work. So I also want to thank Bud for his leadership and mentorship and support all the time, and also Sean for teaming up on so many important NACC initiatives. Okay, let's jump in. So we have made a lot of progress since last fall when I was last up here, and I'm excited to share that with you. So NACC serves as the data collaboration and coordination hub for NIA's ADRC program, as you all know. And we have made a lot of progress across both of these areas. But today, I'm going to be focusing on our data coordination function. I'll highlight some important UDS-V4 updates, share how the new NACC data platform will transform how ADRCs interact with their data, as well as advance our ability to collect, integrate, and harmonize and share multimodal data from across the ADRC program to accelerate ADRD research everywhere. So NACC data is already fair, I want to say that, and we're about to make it even fairer. So yesterday, Bud mentioned that there has been a 61% increase in our data requests and a nearly 40% increase in publications using our data over the past year. And as of last March, NACC data had been used for over 1,100 publications. So we'll be closing out the NACC session today with talks from 3 fantastic researchers who are using NACC data in impactful and innovative ways to advance the field. So I look forward to having Narjez Razavian, Vijay Kolachalama, and Tim Holman join on the stage later. So you can access NACC data today through a new quick access file system and you can check it out for yourself. It takes about 15 minutes to submit a data request. You can get all of our data. It's all free, as many of you probably know, and you will get that data within 24 to 48 hours. So now I want to share more about why NACC data is about to get even better and more fair. So as many of you know, NACC is going through a process of dramatic modernization right now. We don't just want to align with NIH's data science strategy. We want to be the leading edge of innovations happening in that space, and we're striving to be, a shining example of how you can transition from an older data platform to a modern data platform. So we also don't want to build it and hope that you come. So everything that NACC is building, we're building in collaboration with other key stakeholders from across the ADRC program and from across the ADRD research community. And so I want to just really emphasize that. So we are collaborating with the Clinical Task Force on UDSV4. We are collaborating with Electronic Data Work Group on building a streamlined data capture system. You'll be hearing a lot about that from Sudeshna Das later on. And we are working with all sorts of ADRC steering committees to integrate new data modalities. And we are also working with a number of different stakeholders, including NIGADs, NICRAD, a number of different ADRCs, as we're working to develop an advanced data search and access system as well for our data. So I want to say a little bit more about UDS4. UDS4 is going to allow us to modernize and standardize data collection in alignment with advances in the field. Yesterday, Bud discussed how capturing data on these modalities, these different modifying drugs, will allow us to leverage the incredibly rich UDS neurocognitive data, as well as other data modalities within NACC, to better understand the impact of these drugs in the course of the disease. Gil Rabinovich will be speaking about how the inclusion of this data will also allow us to build meaningful connections with studies such as ALZNet. And Lisa Barnes will be speaking about how the new mandatory data collection focus on SDOH. And also about how this new UDS4 will be more inclusive. In addition, there are new concepts that Bud mentioned yesterday as well: subjective cognitive decline, mild behavior impairment, as well as the addition of new verbal learning tests that will be really important for being able to capture important data to advance the field. So NACC is eager to partner with the ADRC community to facilitate more inclusive ADRD research and to advance our understanding of ADRD across diverse groups. We will be collaborating with several ADRCs to provide new tools to support more diverse recruitment. This will include Spanish and Chinese translations of UDSV4. NACC is also seeking to expand our collaboration with the Indigenous ADRD Advisory Group. This group is spearheaded by Yowei Loon McCluster Davis and Jordan Lewis. There will be actually a workshop later today that I know many of you are planning to attend. And NACC has applied for funding to support their work further. That's focused on data sovereignty, as well as creating on-ramps for ADRCs to respectfully and successfully engage Indigenous populations. The introduction of electronic data capture for UDSV4 will also increase access by enabling telecognitive assessments to be taken anywhere in the world on any device. So I want to thank all the members of the CTF for their leadership and collaboration, and Alan Levy in particular for chairing this process. The changes that you're overseeing are going to have a major impact on the field. And I also want to give a huge shout out to Melissa Lurch on the NACC team, who has done an incredible job of shepherding this process along and making sure that UDSV4 is moving forward effectively. So thank you, Melissa. So Sudeshna Das is going to say a lot about electronic data capture in her whole 5 minutes. And the amazing progress that we are making in collaboration with this incredible group. As part of our EDC system, so I'm not going to say much about that, but I am going to add one thing, which is that as part of our EDC system, the NACC team has been working really hard to build transparent QC processes for all of UDSV4. So eventually, all of the QC tools that we are building will be publicly available via GitHub. And I want to give a special thanks to Catherine and Chandima for their hard work on leading that effort. So we'll also be forming, you'll be getting an email about this soon, a rules engine task force coming soon, and you can learn more about that at the UDSV4 EDC booth, and we are going to be looking for volunteers to get involved in that. We have 84 members of our EDC work group so far. So we have built a robust strategic roadmap for rolling out UDSV4. This includes a plan for all the associated QC, EDC, translations, trainings. We applied for an administrative supplement to support Spanish translations, and we'll be finding out about that funding soon. I'm not going to go through all of these details with you, don't worry. But actually, there's a big printout of this in the hall if you want to know more about any of this. And the team members out there will be happy to talk through it with you. And we'll continue from there. So I want to also mention that we will be keeping you in the loop. So we do have a UDSV4 webpage, an EDC workgroup webpage where you can get updates regularly. And we also, of course, will be sending emails as well. Okay, so now I want to talk about the new NACC data platform and data front door. I'm happy to report that we've been making, we are building, as you've probably heard, a multimodal cloud-based data integration platform with advanced data search capabilities. So I'm happy to report that we've made some great progress towards launching this new platform since last fall. I'm excited to introduce you to some of its new features today. So the ADRC portals, so yes, here it is. You guys have seen this slide a bunch, so I'm not going to spend much time on that, but I'm going to start with talking about the ADRC portals, which are a new feature of the platform. So the ADRC portals will provide ADRCs with self-service access to view, download, and audit your data. So these portals will replace inefficient, static monthly reports. NACC has developed an ADRC portal prototype and is actively conducting user testing with ADRC representatives. Each ADRC will have their own private ADRC portal within the NACC data platform that is only accessible to designated ADRC representatives. So thank you to Ben Keller on the NACC team for spearheading this effort. And the ADRC portals will allow ADRCs to access a lot of different kinds of data and metadata. So you'll be able to access your UDS data, you'll be able to access neuropathology reports, non-standard MRI PET. We are going to be launching a participant diversity dashboard where you can see where your participant diversity stands and more on demand. We are going to be having ADRC standard MRI PET scan dashboards, QC dashboards. Beth Mormino will share more about that. As well as it will be a place where you can come and access some of your NICRAD and NIGADS data for your participants. That's something we're working to develop with NICRAD and NIGADS right now. So Beth will be talking about that. And then coming this summer, we're really going to be rolling this out further. So today, you can actually go provide feedback. There's several team members that are in the hall at our booths where you can go and do a demo of this prototype and learn how to get access permissions for the ADRC portals. We also will be giving data managers access to view, audit, and check out their data and provide feedback this summer. And we'll be allowing directors to demo the on-demand report feature and provide feedback. So like I said, everything we're trying to build, we're doing it iteratively with lots of input from stakeholders before we roll out the final product. Okay, so there's also an NIA portal, and this is essentially a roll-up of the ADRC data portals. Has a summary of everything that's happening across all the ADRCs to make it easier for folks to be able to, at NIA, to be able to keep track of how the ADRC program is doing as a whole. So next I want to highlight some changes that all of this data, of course, will be available through the data front door and accessible to investigators everywhere through this several different components that I want to introduce you to. So the data front door is a one-stop shop for all ADRC data for all audience that access Alzheimer's research data sets. So the tool will be freely accessible to researchers around the world via the NACC data platform, and it will allow for data sharing via quick access files, an innovative tool that lets researchers build their own multimodal data sets, powered by Leaf, as well as access to the SCAN public dashboard, which Beth will also share about. I want to give a quick highlight with regards to the quick access files. So this is really exciting. So coming this summer, we will be making available through our quick access files all of the SCAN, SUVR, and volume data for MRI and PET. So this is a big, big deal. This has been a huge effort by the SCAN team, collaboration with NIA, with LONI, with others, Imaging Steering Committee, to make this happen. We are so close to being able to share this with everyone, and that's going to have a big impact on moving the field forward. So the other thing I want to mention is that We also, NACC has collaborated closely with NICRAD, NIGAD, SCAN, ADRCs, and others to begin to define requirements for the data front door cohort selection tool. And we're planning to hold focus groups and conduct prototype testing this summer, so stay tuned for more information about that. And I already mentioned that Beth is going to be introducing the SCAN public dashboard, so I'm not going to say much about that, other than that I hope you all are going to like it. It's going to be useful for researchers. So now I'm going to highlight how integrating these multimodal data streams into the NACC data platform is accelerating ADRD research and discovery. So you probably can't see that very well, so let me zoom on a couple more things here. So I just want to say as a starting point that integrating all of these different data streams and making them accessible through this one portal, we're really optimistic that this is going to be making it possible for researchers to ask and answer new questions in Alzheimer's disease and ADRD, and we are just incredibly excited about that. So I want to make a few, I want to highlight, we're also optimistic that this will allow for more differentiated disease stratification and will be important for improving individualized diagnosis and prognosis of expected clinical progression. We're also optimistic that this new resource will create more opportunities for the development of precision medicine approaches for therapeutic intervention. So lots and lots of exciting things hopefully coming down the line using this data. And we've made especially strong progress, as I mentioned, in integrating standard MRI and PET data through SCAN in collaboration with the whole SCAN team, Imaging Steering Committee, and others. And I'll share more about that at the Imaging Steering Committee meeting later today, so I'm not going to talk about that now. So NACC is also collaborating with several ADRCs to integrate EHR data focused on COVID and ADRD. I want to thank our partners at these pilot sites, so at Indiana and at Columbia, as well as at WashU. So establishing governance for new data streams and new data modalities is actually really hard, and I want to thank them for partnering with us on this effort to really try to solve this puzzle for EHR and claims data. And I'm hopeful that the work that we've been doing together will help pave the way for EHR data collection across the ADRC program. We'll be able to scale that, and also for collecting new data modalities in the future. So I think we really helped pave the way for that. We're also collaborating with NICRAD to explore new ways that we can return ADRC data both back to ADRCs, as well as hopefully eventually more of that with researchers through the data front door. That would just be the metadata and analysis data. All of that data still lives at NICRAD. And you probably all received some data back via REDCap last month. So we're also doing the same with NIGADs. Once again, the metadata analysis data. We just had a major breakthrough with NIGADs through our COVID supplement work, actually, and have finally, I think, solved the puzzle of how we're going to integrate that data as well, the other data streams that I mentioned. So I'm once again hoping that will be applicable to other data streams in the future. And then there's actually one slide missing here, but I was about to circle digital health, and I did want to, the digital biomarker. And so we are, as Nina mentioned yesterday, we are in the process of, Rhoda Al, Nina, and myself have been working hard to figure out how to reimagine the tech work group. So that is about to be relaunched. We are looking for new members to be a part of that. All of the co-leads have already been assigned, but we're going to need lots of people involved to really help move that data modality forward. So if you're interested in that, this should say Rhoda Al's email on here, but it's rhodaal@bu.edu, and I encourage you to reach out to her if you're interested in doing that. And I have an exciting thing to share, which is that NACC will be partnering with the Gates Ventures to essentially roll out a new pilot program. So the goal of these pilots is to accelerate innovative digital biomarker collection sharing across the ADRC program to advance ADRD research and discovery. And we really want to be leveraging technology to capture more data with less burden on participants and ADRCs. So there'll be 1 to 3 pilots ranging from 250K to $1 million each. The pilot lengths will be 1 to 2 years in length. And this will be open to any ADRC or non-ADRC group or company. So they must have multiple ADRC collaborators. So funding will be channeled through NACC and will support partners in providing digital instruments. We'll support ADRCs in doing that data collection with innovative digital instruments. And NACC will get some funding to integrate this data into the NACC data platform and share it with ADRC researchers through the data front door. ADRD researchers, apologies. The selection committee is TBA. There's some important requirements here. So it has to add research value and enhance metrics. It must reduce burden. It must expand and address diversity. It must involve multiple ADRCs. And we must demonstrate scalability. It must demonstrate the potential to be scalable to all ADRCs in the future and be amenable to data challenges down the line. And also partnering companies, this is really important, must be willing to provide the raw digital files back to NACC for sharing with everyone. So we're interested in lots of different modalities. You can see those all listed here. Interested in all sorts of tools. You can see the target timelines there that we're going to be working with. So the goal is to accelerate this process, right? It's faster than applying for a grant. We want to be innovating in the digital arena much more quickly. So that's why we're doing this. So lots of potential follow-up support, too, for successful projects, potentially getting some funding through for a big data challenge, to run on the data that's been collected. We're going to be looking at promising digital pilots to try to include in the NACC renewal. And there's also lots of opportunities for SBR and SCTR funding, potentially for some of these projects, as well as other means. So we're really excited about this and hope that lots of people apply. So the last thing I want to say about our platform is that we're expanding multimodal data integration, as I said. We're really making access to this rich multimodal data, creating new opportunities for that. And this platform that we're building is scalable to new data streams. It allows for this real-time data search, visualization, access, and it's ready for AI-driven discovery. And you're going to hear how there's already folks using NACC data for AI in really interesting ways, but this is going to really ramp that up. So we're also looking to build interoperability with other data repositories. So we're working on a collaboration right now with the AD Knowledge Portal at Sage to work on integrating ways that we can integrate their data as well. And I want to share some of the challenges. So creating governance, I already said was hard. That will always be hard. It takes, we're going to be updating the process by which unique identifiers, as NACC IDs, are assigned at ADRCs, so stay tuned for that. That's really important as we start to add new data modalities. We're going to be building pipelines to collect and integrate new data streams. That can be hard as well. We want to be optimizing search for each of these data streams. And we also, as I mentioned, want to be building interoperability with other repositories, so some challenges there. Future opportunities. So we want to integrate more real-world data. We want to build seamless interoperability with these other platforms that I already mentioned, as well as future ones. We want more digital health and more data with less burden via those tools. And we want more languages, more diversity, more access. We want to reduce barriers to data access and analysis by providing secure enclaves. That's something we can do with our current data platform. Don't have the funding to do it right now, but hope to in the future. This is another roadmap of kind of how we're going to be rolling out different components of our new data platform. You will see a lot of things in orange here. So some of these things are things that we have applied to continue working on through an administrative supplement, which we'll be finding out about soon. We're very hopeful about that will really help us to stay on track with being able to deliver on these exciting new tools, both for the ADRCs and for the ADRD research community. So, like I said, we don't want to build it and hope that you come. So please, 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 there's all these wonderful people sitting at booths out there. They have demos for you. You can go check out how the skip logic is going to work in these new REDCap forums. You can give feedback on the prototype for the ADRC portals. You can actually touch and feel these new UDS forums and ask questions of folks. And you can also learn about our amazing NACC directory. The mastermind of that directory, Laura, is right back there. And she's at that booth, and she's going to be excited to talk to you about what she's built. So thank you all very much, and I want to call up the next speaker. Okay, Lisa Barnes, thank you.
Good morning. How's everybody? I want to thank NACC for inviting me to be in the hot seat once again, and Megan Zilsdorf, my co-chair on this SDOH module. So I want you all to remember the fall. Everyone was so excited about social determinants. It was electric in the room, right? Hold on to that as we go through this 5-minute presentation. So why are we including social determinants in the UDS-4? I have talked about it at least 4 times since the beginning of COVID, but for those of you who don't remember, social determinants are conditions in the environment where people live, learn, work, and age. We know that they impact health, including brain health. It will help us understand heterogeneity of cognitive aging, We know that social determinants shape health behaviors and they interact with each other to produce health. Really, they're just too important to ignore. I hope that's really, really clear by now. So, as a reminder of the process, we had a committee of 13 content experts representing 10 ADRCs. We worked for about 6 months to develop the forms. Then NACC disseminated 2 red cap surveys to collect feedback on the content from all of you. We got the surveys back, got 46 respondents from about 31 ADRCs, 120 comments. I think that's the most ever of any survey. We listened to the feedback and we incorporated it, and so now here's the update. So you might remember that there were some social determinant questions in Form A1, which is the demographics form. We removed those questions to eliminate redundancy and improve efficiency, and we incorporated them into the social determinants form, A1. So now this new and improved form is about 35 questions. We didn't change the constructs, we just now have an extra construct, discrimination exposure, which was originally on the demographics form. So now this form, the exciting part is that it's mandatory now. It used to be optional, but you were so excited. You wanted this. So we had to listen. So now it's mandatory, but it's only one time. So don't worry, it's not going to be a huge lift. We want it to be self-administered. We recommend it be self-administered by people with a CDR of 0, 0.5, or 1. If you want to go out further, by all means, you can do that. We did test this in some, pilot testing, Megan and I, in Wisconsin and Chicago, across a range of people, and we think it takes less than 10 minutes if it's self-administered. If you have to administer it, it might add about 5 minutes. So there are other exciting opportunities to collect health equity data in UDS. We always had the ADI on that Form A1 that's still there to measure neighborhood disadvantage. We're going to ask about the participants' primary occupation throughout their working life. So this is all going to really expand our look into how these determinants are impacting health. We also have a number of questions focused on gender and race, so we can be more inclusive. We also ask about sexual orientation. So we ask about gender identity, sex at birth, intersex, and sexual orientation. And NACC aligned these questions with the questions from the national academies to ensure the best representation. And we're much more flexible in listing multiple racial identity. We're not forcing people to only say, I just have 3 races. People there's a wide variability in how people identify. So now you can have an unlimited number of racial identities. And you can learn more about these exciting changes at the booths out there. So please do go visit and look at the questions and see how they're working for you. And dropping today, we have a DEI-focused Dementia Matters special series, starting off with Sarah Biber today. This is the Dementia Matters series that comes out of the Wisconsin ADRC. And it's Dr. Chin's baby, so hopefully you'll listen. You can scan this little cute barcode to get all the dates. You'll see that after Sarah, I have my interview, then Gina Green Harris from Wisconsin, and then our very own Crystal Glover from Rush. So thank you, and looking forward to the Q&A.
Okay, next up is Beth Marmino from Stanford ADRC. Thanks, Beth.
All right, there's no jokes about that light. Okay, so I'm going to talk about standardized imaging in the ADRCs. Okay, so I think we're quite excited about standardized imaging. It's been there's been some growing pains throughout the centers, but I think, we're really at a transition point. So just as a reminder, amyloid and tau PET imaging, along with MRI, since 2005 has really become central to many ADRD research efforts. However, uniform imaging was not routinely captured in a standardized way or shared throughout the ADRC program. So in 2017, there was a work group including imaging core leaders or biomarker leaders involved with ADRCs and also directors that met to discuss this, and along with NIA and NACC, and essentially recommended that we try to transition as a program to having a standardized set of PET and MRI data throughout the program, and that was also uniformly shared. So in response to this, in 2020, the SCAN initiative was funded. So this is led by Bill Jagus and Cliff Jack, along with a very large team. And their goal is to really help the program to promote this standardized acquisitions, to analyze this data, and ultimately to make it available to the scientific community. In 2021, so just 2 years ago, the ADRC sites started contributing their standardized imaging to SCAN. And what we're going to be seeing over in this year is actually the first sharing of this effort that really has been stretched out over some time. So this is what the current infrastructure looks like. So the individual sites are collecting the data. That's been going to LONI. SCAN has been analyzing this data. And it's just about to be released. So I want to spend a couple slides, talking about how NACC has really, helped, this infrastructure, helped us with this transition, and really enabled many aspects of this really exciting program. So this plot is showing on the y-axis the number of standardized PET scans that have been given to SCAN from the individual sites. Time is on the x-axis. So as we know, we started giving this data in 2021, and at first it was very slow. There was it was kind of a slow slope of data contributions. So one thing that NACC did for us is they developed these checklists to help us understand what do you actually have to do, to what sort of certifications, qualifications you have to go through, how do you actually upload the data. So they developed that for us. And they also went one step further with NIA. They ended up reaching out to the individual sites, conducting interviews, understanding what are the actual barriers to completing this. So that's shown in the yellow rectangle here, and you could see there's a significant increase in the slope of uploads after that event. So NACC really kind of helped us. This program was in place, but really accelerated our ability to participate in this program. So Sarah mentioned this. There's two different types of dashboards that NACC is developing for us. So the first is a public dashboard that really highlights effectively the readiness of each individual site to participate in SCAN. It also lists the number of participants that SCAN data has been collected on, and then by modality. So this is really a tool for us to understand the scope of this program and the contributions across all the sites of actual SCAN data for both PET and MRI. Another dashboard that NACC is developing for us is a private dashboard. So this is more detailed information at the site level. So it's been, from the site's perspective, it's been difficult to understand. We're giving this data, and we don't know if it's okay, what the QC status is. So this private dashboard is going to be kind of collective. It's a record, effectively, for each site of all the individual uploads that have happened and the status of those uploads, whether or not they've passed or not, and moved on to analysis with SCAN. Oops, I went too far. Okay, and so we've heard about this today and also yesterday, this concept of the data front door. So this is, in the future, NACC is going to be working on this, and it's really exciting because there's all these amazing modalities that have been collected on NACC participants, and this is going to really bind them and enable investigators to understand what is out there and how to best use this data. So the point of this slide is just to highlight that now the standardized imaging is going to be a part of this platform. It's going to be able to be linked to all this other exciting data within NACC, I mean, there's some very basic questions that investigators have about NACC data. You know, what type of data is available? You know, how many PET scans are there? How do I access this data? How do I get approval, you know, for these different types of data? These are key concepts that this front door is really going to help the scientific community with data access. And so this is a long-term goal, but I just want to stress, you know, the immediate outcome of this effort with SCAN is going to be the sharing of the actual standardized extracted values. This is going to be available very shortly as part of the quick access file with NACC. So we're really going to be able to start using this data for the first time. So I'm going to conclude with a few statements. So it's been a long road to get to this point, but the progress, I think, has been quite impressive. So just in this 2 years of standardized image capture and collection throughout the ADRC program, there's already 900 amyloid PET scans that have been analyzed by SCAN. There's over 500 tau PET scans in just this 2-year process. You know, this really reflects a very, I think, productive and engaging collaboration between the individual sites to go through this. This has been, you know, it's quite the transition for us in collaboration with NACC, SCAN, LONI, and NIA. There's a number of immediate milestones that NACC is going to be helping us with. So the dashboards, which I told you about, NACC's just started returning the individual SCAN data to sites, in part to check to make sure the data is valid, but that's going to be something NACC is going to be doing for us. And the ultimate goal, I think, is to share this exciting data with the scientific community. You know, I think there is going to be quite the impact of this new standardized imaging data set. The ADRC program is quite exciting in that it's a large multi-etiology cohort that has extensive other data types that's already collected and already associated with these participants. And another thing I'm very excited about, this is really, you know, this is kind of a 2-year, almost a pilot in a way. This is just the beginning. You know, we already know in the new RFA, there's going to be more imaging, you know, more standardized imaging across the sites. And then, you know, we're crossing our fingers that Clarity gets funded too to allow us to address some scientific aims related to multi-etiology and heterogeneity in the ADRC program. So thank you.
Thanks so much, Beth. Next, I'd like to call up Sudesh Nadash from the Massachusetts ADRC.
Thank you, NACC, for inviting me to do this talk. And it's been my distinct pleasure to work with Dr. Sarah Biber to launch and kick off and see the electronic data capture working group flourish. It's a NACC and ADRC collaborative effort. And it's kind of hard to believe, but some of us get easily excited about collaborative infrastructure work as much as science, but so bear with me. The goals of the Electronic Data Capture Working Group was to develop a modern EDC solution to effectively serve the needs of the ADRC program. It was to foster collaborative software development, build robust and scalable solutions. You know, modernization has been a theme that you've heard from Sarah and throughout this entire session. To build a data community across the NACC and the ADRC so that we could work together as like one community to promote and share best practices and to make data-driven decisions with user surveys. The electronic data capture system was born out of the Data Core Steering Committee, and its particular goal was to address the needs of UDS version 4. And it was launched by NACC and the Data Core Steering Committee in January of 2022, when we felt that UDS version 4 was imminent within 6 months, but you know, exciting to see all that progress. And really exciting to see that now we have like 84 people across 32 ADRCs that are collaborating across these different subgroups of the Electronic Data Capture Working Group. And there's been like diverse representations from like 32 ADRCs, including 2 new ADRCs and 3 exploratory ones. And you can see on this map here that it's really, we've had like wide representation from, you know, junior staff members to like core leaders and part of this UDS version 4. So the electronic data capture working group has 3 different components to it: the requirements subgroup, the development subgroup, and the documentation and training subgroup. The requirements subgroup, as the name reflects, it collects all the requirements and harmonizes those across the different ADRC programs. The development subgroups has actually been involved in developing the forms, and the documentation and training to build SOPs and protocols so that those can be widely shared as a community resource. And all the work for the electronic data capture working group for UDS version 4 has been centralized around REDCap forms. So that is a technology that we adopted after doing surveys, because most of the ADRCs either were already using the REDCap forms for their electronic data capture systems, or were interested in transitioning to the REDCap form. There's some hesitation for especially those centers who have had large amounts of data, and it's hard to migrate all that years of data into REDCap. But something that we can think about is that you can still have these REDCap forms that we are developing as your EDC, and then store them into your legacy system. So it's like some kind of like minimal effort. And the data capture working group is here to help in those transitions. And the other advantage of REDCaps is that these are available on mobile devices, so they can be deployed by and used in iPads in waiting rooms or really anywhere in the world. The 3 work groups have made tremendous progress over time. The requirements subgroup determined the common UDS data flow used across 29 participating ADRCs, and there was the survey was sent to all 37 ADRCs, and we got really near full response. They deployed the data management plan in alignment with NIH's data sharing policy, which we heard about yesterday from NIA. And it's really been a very valuable resource for ADRCs seeking renewal, including ours. They summarized data quality checks for UDS version 4 to be shared with all the ADRCs. So as Sarah mentioned, there'll be an actual rules working group as well. The development work group really did a tremendous amount of work to streamline and standardize the REDCap submission process, implemented the quality checks and variable names. They established infrastructure for longitudinal data collection in REDCap, and 40% of the REDCap forms for UDS4 have been developed, and review process is underway. So this collective effort can be now, all the other ADRCs can share the fruits, and when UDS4 comes along, each one of us is not left to be implementing these forms all by ourselves. So the documentation and training subgroup also had a lot of work. They built a curated directory of valuable REDCap resources, especially useful for those new or other ADRCs who are looking to transition to REDCap. They developed NACC-specific REDCap SOPs to facilitate and help these ADRCs, and designed a web page to make all these resources readily accessible to the ADRCs. So all our work and having a common electronic data capture system has had some really practical benefits. It has helped in streamlining and reducing the burden with electronic data capture, other than being able to, to have a harmonized and set of forms. It's helped in specific examples of how it's saved time. There are autofill drug options from RxNorm, so that's been integrated. There's auto-populated health history data for all the follow-up visits, so you don't have to copy and paste. There are skip logics, access to automated NACC tools, and then on-demand centralized robust training as people are actually, the doctors are using the forms for electronic data capture. Doctors and anybody else who is entering data. These forms have also helped in enhanced sharing, improved solution co-development and sharing among the ADRCs. It's conserved resources through centralizations, do it once, share it with others, rather than each ADRC building their own system. And then we've had some little progress in automatic EHR extraction from Epic. That ADRCs who capture EHR for UDS data visits can take advantage of. And then, of course, electronic data capture, much as we love our manual processes, does help improve data quality because it provides real-time form error checks, reduces multiple choice errors. There are like directed guides. It provides a transparent and replicable QC process, improves standardization, and enhanced utilization for analysis and other scholarly activities. So this makes the process where the ADRCs who are depositing data and the ones who are the over 1,000 researchers who access the data, there is a minimum amount of like translation and changes that are going on between the data capture, transmission to NACC, and then out through the other way. So I'd like to thank all the members of the electronic data capture work group who have made this tremendous work possible. First and foremost, Dr. Sarah Biber, who has helped give birth to this team and has been leading this effort. The development co-leads, John and Melissa, the requirement co-leads, Meredith, Chad, and Carrie, the documentation and training co-leads, Sarah and Laura, and Hannah, who has been involved in coordination. And really a big shout out to all the NACC members who have taken on all the logistics of creating such a working group and made our lives easier. And it's always nice to see their smiling faces over Zoom. So thank you very much.
Thanks, Sudeshna. Next, I'd like to call up Gil Rabinovich from UCSF, ADRC.
Good morning, everyone. Really a pleasure to be here to talk about potential collaborations between NACC and the Alzheimer's Network, or ALZNet. So ALZNet is, for those who aren't familiar, is a national network where clinicians will enroll patients who are being evaluated for treatment with novel FDA-approved AD therapies, and they'll follow them longitudinally to measure their cognitive and safety outcomes. The registry is really agnostic to any specific drug or mechanism of action, so it will be enrolling people who are being treated with so-called disease-modifying therapies, but also for drugs that are enhancing cognition or targeting behavioral or neuropsychiatric symptoms. ALZNet is really a real-world study, so it's not meant to be a clinical trial, and it's mainly meant to record what is happening in these real-world practices to patients who are being treated with novel drugs. And really, the current version of ALZNet is just a first version, and this will be sort of a living protocol that will evolve as therapies evolve in the field. This is really a very large team effort. ALZNet is led by the Alzheimer's Association, partnering with the American College of Radiology and the Brown School of Public Health. This is the team that also has been leading the ideas and new ideas studies. In addition, we've partnered with the American Society of Neuroradiology and the Critical Path Institute. The ALZNet is led by Maria Carrillo, Mike Raffi, and myself, and you'll see a lot of very familiar names from ADRCs in our list of study investigators and advisors. Also, a big shout out to Heather Snyder and Rebecca Edelmeyer from the Alzheimer's Association and Andrew March from the ACR, who have been really working tirelessly on this effort. So the goals of ALZNet are first and foremost to develop this network. Now, recognizing that we are really moving into a new era, of clinical care, ALZNet is also designed to help build and implement resources. Oh, goodness. I think I got my old version of the slides, the one with the timer. Can you pause this slide, please? Okay, I'm going to be struggling with this. So ALZNet is going to help provide education and resources for sites that are really learning as they are beginning treatments with these novel therapies, really beginning also to learn how to use biomarkers for diagnosis or to implement molecular-specific therapies. The main goal of ALZNet is to collect patient data at the beginning of the study and to follow people longitudinally, collecting data about functional, cognitive, and safety outcomes. And also, ALZNet will be collecting neuroimaging studies, collecting biosamples, biomarker and genetic data, following individuals and identifying health outcomes through Medicare claims and other databases. And very importantly, a major goal of ALZNet is also to share these resources with the research community so that all of you can really use this real-world data to try to understand the disease better. So the study has launched with 9 Vanguard sites. Really, the limited availability of the new FDA-approved therapies has limited the number of sites and patients that are currently being treated, but we are really in a rapid development phase right now where about 100 sites are currently being either actively in startup or are being evaluated as sites. Many of these are ADRCs, and we very much hope that the ADRC network will be all the ADRCs here will be interested in participating in ALZNet so that we can continue to follow your patients. Thank you. Hopefully this will work better now. So here you can see our current progress report. So really, since the very early days, we've been in touch with the ADRC leadership, with Drs. Silverberg and Elliott, to think about opportunities for collaboration between ALZNet and NACC. And there are really many potential opportunities, I think, for, first of all, enriching phenotyping. So I think one of the lowest hanging fruits is co-enrolling individuals in NACC and ALZNet. Now, ALZNet is really meant to be implemented in clinical practice in a very seamless way. And so the only required instruments that are collected are either the MMSE and MoCA and the FAQ. 88 and NPI are optional. So you can see in bold there is some overlap with UDS, but of course having individuals co-enrolled in NACC will allow us to get full UDS, to leverage the amazing infrastructure that's been built around genetics, biomarkers, and research-grade neuroimaging. And also, very importantly, not on this slide, but mentioned several times yesterday, the opportunity to follow people who are being treated with these novel drugs to autopsy and really understand at the tissue level what has been going on in the brain for people who have been exposed to these drugs. UDS will be collecting data on new drugs, but ALZNet will be collecting this in a more detailed fashion, also collecting data on adverse events. And so there's a real opportunity, I think, by co-enrollment to enrich both studies through this deeper phenotyping. Having common data elements will enable comparisons between the two studies. Of course, it doesn't simulate a randomized clinical trial, but just as an example, there'll be an opportunity to compare treated patients in ALZNet to similar people who are untreated and enrolled in NACC, contemporaneous or historical controls. And I think the real power here is to take a study like NACC with individuals that are followed at specialty centers like the ADRCs and then use cognitive and longitudinal trajectories or predictive models that are developed in that kind of cohort and try to validate them in a real-world cohort that is represented by ALZNet in order to really see if some of these models and trajectories that we're studying in NACC do translate into the real world. There are also, the devil's in the details, of course, so there are also a number of things we need to figure out. I think one is the difference between clinical care and research. So ALZNet activities are part of clinical care. ADRC visits, of course, are observational research. I think at some ADRCs, there will be a seamless integration between the clinic and research, and in others, this may be more of a challenge. So this is something that we need to figure out. Of course, a major goal will be to minimize subject and site burden, so any instrument should just be delivered once to a subject and then hopefully recorded in both databases. And then, of course, the integration and linkage between the databases that are quite different is a major challenge, but I think very much in line with what we heard from Sarah and others in terms of what NACC is currently doing. And so stay tuned this summer for another NACC survey. I'm hoping for as much enthusiasm as you had for Lisa's survey, maybe not, but hopefully we'll get good return because we're very much interested in your input in terms of how to address some of these issues. And we will be also holding a webinar about ALZNet NACC collaboration later this summer. Of course, ALZNet is really just taking provider-entered data, and there's a huge need to really scale up real-world data in our field, electronic health records, patient and care partner reported measures, linkage to other existing cohorts and studies. Again, we heard this is very much in line with what NACC is already doing, so we were all very excited about the announcement of the ADRD real-world platform U54 funding opportunity. I think it's no secret that the ALZNet team will be applying for this and that ALZNet will be just a small piece in this major effort to understand real-world ADRD care. And so this is the barcode. We're hoping, again, that all ADRCs will be part of this. Please scan this if you have not yet applied to be a site. And apologies for the technical difficulties, but really thank you for your attention and looking forward to answering your questions during the Q&A.
Thanks so much. Next, I'd like to call up Vijay Kolachalama from Boston University ADRC.
Good morning. The last time I was here, I was asked to give a 5-minute lightning talk. And today I am talking for 10 minutes, so that's upgrade. Thank you, Sarah. So before I talk, these are my disclosures and funding agencies. So I think we talked about team science briefly, and I want to just sort of really highlight this slide a little bit because this is my proudest achievement. And I think you can see here, based on the unique colors I have chosen, there are people with different backgrounds in my lab, and I'm very proud of it because I think when I bring people who are doing computer science, who are getting their PhDs in computer science, along with some MD students, as well as some other trainees who have some experience in neuroscience, magic can happen. Because all the data that NACC has been collecting over many, many years, we have been the direct recipients of using that data and the beneficiaries of trying to think about methods. So here I want to give a particular shout out to the computer scientists. They're lazy programmers, right? They don't like to write long codes. And they kind of really also try to figure out a way to sort of skip work, right? So, you know what I mean? It's not like they're like really lazy, and that's also there, but you know what I mean? They don't like to do a lot of programming. Actually, you know what? Let me revise that. So they're actually very efficient programmers. They write very smart programs to sort of make things easy for me, right? So that probably came out well. Yeah. So anyway, so we just started to work with NACC data, and I think we sort of realized that there are lots of interesting questions that we can ask. And I think one of the first things we started to ask was a very simple question, which is based on, I think, one of the papers that I saw about 5 years ago. It's focused on talking about the supply and demand in the neurology practice. That kind of really inspired me because you're collecting all this data, and at the end of the day, some of this data is kind of routinely collected in a clinical setting. So the simple question is, can you sort of really build AI approaches to sort of really use the routinely collected data and then sort of provide some kind of a screening tool for dementia assessment, right? That was one of the first questions we asked. And we don't want to just build computational models. We also want to make sure that these models are validated in a nice way. So we sort of spend a significant amount of time to validate these models. So we bring all these experts, clinical experts, to sort of help us with this head-to-head validation. And for some cases, we also do some kind of post-mortem validation, right? So this is the NACC data that we downloaded maybe 4 years ago, or maybe 3.5 years ago. I mean, you see the number is only 4,800. I know there are many, many more cases, Sarah, but this is the data we downloaded. And I think the first question we were trying to ask is, okay, given this kind of a multimodal data, which means we are talking about data that is basically neuropsychological testing, demographics, patient history, imaging, can you basically take all this information and can you try to ask a simple question, which is, can you predict if this patient has, or this person has healthy cognition, MCI, or dementia? And if this person has dementia, can you sort of really point out whether this is due to Alzheimer's or some other etiology, right? That was the question we asked. And we actually spent a lot of time to sort of process this data, even though there are a lot of efforts to collect the data. Once you actually have the data, there is a lot of work to do after that. So I want to sort of briefly talk about it. And here is a list of all the things we do once we collect the data, right? So we spend a significant amount of time. So the green bar that shows actually the time spent on each of those tasks. So it's about 75% of the time after we download the data, we actually spend to think about automating these things for all those 4,000 cases or 4,000 plus cases, to think about registration, normalization, segmentation, selection of regions, bias correction if there is multimodal data primarily, and then sort of do some kind of image quality check. And after that, we think about deep learning, right? So there's a lot of processing work even after we have this data downloaded. And then once, and just again, to give you a quick overview, so on the top you're seeing this kind of a registration process. We sort of created these automated pipelines to sort of register these images, every plane by plane in all 3 different planes on all those thousands of cases, and then also make sure that these things are also looking good, right? So we spend a lot of time to do this image registration, and then also we come up with automated routines to sort of remove that bright thing around the brain, and then make sure that this data is actually amenable for machine learning, right? So once we do all this, we actually were able to publish some work, and these are the couple of papers that we recently published. And again, just to go through this whole idea of thinking about multimodal, right? So we're talking about demographics, we're talking about patient history, talking about functional assessments, neuropsychological testing, all those different forms, A forms and B forms and C forms. So we collect all this data. And then we sort of really are asking this simple question, can you assess if the person has healthy cognition, MCI, or dementia, and if dementia, is it due to Alzheimer's or due to other etiologies? And fortunately, we've been also collecting data from other cohorts, as you can see here on the right. Of course, NACC is the one that's highlighted, because we use NACC for actually training the model, and then use the other data sets to actually test the model. And just quickly go through about how we sort of really process this multimodal data. We sort of take these images and we run through these methods called as convolutional neural networks, which are basically allowing us to process voxelated information in this 3-dimensional sense to sort of hierarchically learn from all the different patterns on those images. And then we sort of extract some kind of information from all these volumetric data, and then we combine that with other forms of data to sort of really ask that question about trying to assess if the person has NC, MCI, or dementia. And once we do that, we are able to sort of go back into those frameworks and sort of really rank order the importance of which features were actually influencing the output of model output performance, right? So as you can see here, the left side is this question about whether the person has healthy cognition, MC, or dementia, and we are able to sort of really rank order the important features that were influencing the output of interest. On the right side, you're seeing this binary question, which is about Alzheimer's versus non-Alzheimer's dementia. And interestingly enough, the first factor that turned out to be important was actually MRI scan. And then once we do that, we also want to go back into those MRI scans to see which regions in the MRI were actually influencing the output. So we have these frameworks that we developed where we can do these kinds of interpretations on how the model is actually trying to assess the output. And that's the advantage of creating these interpretable machine learning approaches rather than thinking about them as black box approaches. Here we're able to go back into those images and see which regions in those MRIs were actually trying to predict the output of interest. And then after we did this, we also did some neuropathology validation. So we had access to some postmortem data, especially those ABC scores, and we are showing here that the model was sort of really, the risk predicted by the model was sort of proportional to the degree of severity that was observed in neuropathology. And you're seeing here A, B, C scores. And then as you can see, there is sort of this increasing trend on the risk that the model predicted, and that sort of was proportional with the pathology. And then finally, we were able to sort of combine the neuropathologic evidence as well as the interpretability to see if the regions that were highlighted by the model also were actually the regions that had high degree of pathology. And this is kind of a nice way to sort of really think about how to sort of create visualizations that will allow us to not only just tell that the model is doing a good job, but at the same time show that the model is identifying regions that also have some kind of neuropathologic evidence. And then finally, icing on the cake, we recruited a few clinicians, and then we asked them the same question. We gave the same NACC data to them, and they say, you know what, can you basically assess if these people have the same kind of aspect, which is healthy cognition, MCI, dementia, and if dementia is in Alzheimer's or non-Alzheimer's dementia. And we had about 17 wonderful neurologists who reviewed all those cases and then gave their assessments. And then on the bottom, you see 7 radiologists who also just looked at the MRIs. They didn't want to predict if somebody had MCI, though. They only wanted to look at the MRIs. So we gave them the MRIs, and we asked them this simple question as to whether they think if the MRI was indicating things about AD or non-AD, and then they kind of gave their assessments. And as you can see, they were not actually very happy with the way they performed. And then, yeah, summary finally, I think we were very fortunate to get access to multi-modal data from NACC, and we were thrilled to sort of really combine all this information to simply ask a question that I think is probably very obvious to this community, but I think that would allow us, that at least allowed us to really think about creating this multi-modal framework to sort of really lay the background in terms of how to think about combining different forms of data. And now that we have this framework, we also want to create a web-based tool, which means you can actually go in and then enter this data and see if you can get the predictions right away. And we are working with multiple different centers because all these neurologists, they're working neurologists in different centers. They're very happy to collaborate with us. And I think it's importantly allowing us to validate our models because we don't want to be just computer scientists. We want to make sure that all the models we are trying to develop actually has some clinical value. I'm learning more in these conferences that there are more questions that we can ask, more exciting questions we can ask, but hopefully this is sort of really setting us the background. Thank you.
Next, I'd like to call up Narjaz Razavian from the NYU ADRC.
Thank you so much. I'm thrilled to be here, and I represent some of our team's work from NYU ADRC on using this amazing data. I've always been excited about NACC, and I am overly excited now after I learned all the good stuff. Go to the next one. So obviously I don't need to repeat this. There is clearly quite a lot of, let's call it disparity or differences in terms of vulnerability to dementia across different racial and ethnic groups. We already know 2/3 of the Alzheimer's patients are women. 30 to 40% Black and Hispanic patients are less likely by 30 or 40% to visit neurologists. These diagnoses for them are happening either in primary care settings. It's not really happening many times in the research cohorts or in advanced care settings. And so there's definitely a lot of need for looking into this data at the broader scale, and I'm really excited about how NACC is moving forward with inclusion and diversity. Early detection obviously is matter. That is now the focus of all of our efforts, starting treatments early. There are also even, without the treatments, there are some trials that actually show if you have cardiovascular burden, you can actually do something if you're at risk or if you're just before transitioning to MCI. So all of this really urges me to improve, put my efforts into improving early detection, especially for these populations who are actually at higher risk for dementia. And obviously, it will also translate to better clinical trials with quite a lot of efforts, collective efforts. We look at NYU Langone Alzheimer's patients, and the second and third column may be of interest. If we just look at overall, diagnosis records, there has been 40,000 cases with diagnosis of various types of ADRD Subtyping is kind of hard in the clinic, so we stick to ADRD as the whole. And then when we look at who got to Barlow Center, less than 10% of them go to Barlow Center. So our goal, I guess, in our group is to utilize power of the data from these, initiatives to, instead of bringing the patient to neurologists, they are not going there for various barriers, scale, accessibility, etc., actually bringing tools to environments where they are getting their care, like PCP centers. So that's kind of our goal, and really our team's focus, focusing on real world, more diverse, more realistic. Obviously, utilizing this data is the key to address this question. And structural MRIs turns out to be an accessible type of data. As we just learned, there's quite a lot of value. In fact, those are among the top predictors. They show atrophies, so that might already be quite helpful. And so with this goal in mind, to go to the clinic to where patients at risk are, we want to focus on accessible modalities like structural MRI, T1 MRI, basically, and ask some questions. Can we detect this? How well can we detect it? How well do these models generalize to different cohorts, large cohorts? And then actually, we are excited that we are also looking now into actually deploying those tools and seeing in reality what happens. Do these models actually really work even in real clinical settings? I won't talk too much about that part, but we utilized the ADNI and NACC data. As I said, I was amazed in 2018 when I logged into the server and I was like, wow, okay, these are all the data queries that I can make, and the data was actually truly, in 2-3 days, I got all the data on Amazon bucket. I love that. And so this study that we are talking about is up to the year before COVID. 2019 is when we got this snapshot of the data. Right now, there's a lot more amazing data. So just a quick note, in 2020, we had to drop everything in my team to focus on COVID, help NYU, and then we kind of resumed that almost a year later, and then kind of we're back to our what we were doing before. But definitely a lot of exciting activity. This data is, let me go back, this data at the time gave us about 15,000 individuals from the NACC, and about 652 individuals with quite a lot of longitudinal scans from ADNI, which was the basis for our data. As we talked about, the criticism is that these cohorts are still a little bit more white than what is reflected, especially in New York City, which is a very diverse city, but also in general. But we started with validation questions. So first, let's build a model and see how well they generalize to external validation. I'm not going to talk about the AI part. We definitely had to innovate in terms of the model themselves. A lot of times people just directly import things from the AI world, but no, you actually need to make some adjustments. And we did that. We published that in a computer science conference, but then we focused on validation of it and actually training it on NACC, and then training it on ADNI and using entirety of NACC as our external validation. We like NACC a little bit better because it has less exclusion criteria, so the disease burden, comorbidity burden of participants are a little bit closer to what is in the real world. We're still talking about research cohort versus real world cohort, but NACC is definitely one leap forward for us, and that's why we wanted to use that for validation of our analysis, and I will show you how the performances are. We can talk more, so which time I have. So ADNI, we split in across different patients, and of course, in cohort validation, gave us really decent result for detecting CN, which is early stage versus the rest, and also detecting AD versus the rest. The performances are pretty good. And we were very happy to see that it also actually translated to really good performances on NACC. When you go to clinic, you kind of act upon certain thresholds, and at that level, PPV and recall are a lot more meaningful metrics, because PPV translates to how many times you show something to the clinician, it's an error, and whether or not they will be discouraged or not. So when you look at PPV of results, let's say on NACC data, this is that's the moment when I said, okay, we need to go to the clinical data now because these are pretty decent. Maybe we can do something with that. So we also compared to FreeSurfer. I guess some know it's FreeSurfer is amazing. It is very slow. At the time, it took us 11 hours per scan versus deep learning model, which, okay, 7 minutes, 7 minutes of pre-processing and less than 7 seconds for the actual model to run. So I was discussing earlier that we want to move towards a model which the moment the patient is in the scan, it can run, it can make some assessment. If there is a need for patient to do anything, they're already right there. Let's get hold of them while they're there before they go home. Anyway, so these are some benefits of having faster tools. Even when the model makes an error, it actually turns out that those patients do transition faster to AD, so perhaps our definitions are a little bit, maybe the boundaries are fluid perhaps, so maybe this model still is useful, and we can look at the performance across different subcohorts, and the performance seems to be, the generalization is there, it's promising, there are quite a lot of encouraging results from our analysis. Also, as I said, so a lot of risk factors for dementia in real world are cardiovascular, midlife cardiovascular issues, right? So NACC allows us to actually evaluate on very large, relatively large subcohorts, people with hypertension, diabetes, various kind of cardiovascular performance, how do they, issues, how do they perform? So this is, again, another reason that we are very excited, and we will continue to analyze and validate our models or redevelop them on new NACC data. There is also potential for explanation of AI, which it's quite useful when you want to interact with clinicians. So now those precision recall curves were the moment where I said, okay, I need to get NYU data because we seem to be onto something on the research data, so we started to pull the MRIs corresponding to 10 neurologist specialists at NYU Langone because we wanted higher quality diagnosis. And we got about 5,000 patients, and they provided quite a lot of rich EHR data and MRI data. And so we validated the model as is, and then just fine-tuning the last layer of it. These are some details, but the performances seem to be encouraging. So that's where we are. It's still in progress in terms of validating it in the clinical setting, but we are really excited. That's our drive, okay, to make these tools available to our entire healthcare community. The model is completely free. You can download the trained model from the website and plug in your MRI. You get a score. And our next steps are actually validating it prospectively in the clinical setting and even running RCTs in the clinical settings. Thank you so much.
Thank you. All right, I'd like to call up Tim Holman from Vanderbilt Exploratory ADRC.
All right, good morning. Very excited to present with you guys. I think we're running a little behind, so I'm going to try to catch us up. Let's see how I can do. Really excited to talk to you about the ways that we're using NACC data to look for precision medicine models of Alzheimer's disease, focusing on risk and resilience in particular. I'm going to skip through this because you guys know it. You heard yesterday from NIGADS and about the Alzheimer's disease sequencing project, about the Alzheimer's disease genetics consortium, from Dr. Farood about NICRAD, and the ways that NACC data has really advanced the field of genomics in the last decade. The largest studies now have included over 1 million people, and NACC has been an international leader in those efforts. You've also heard about molecular biomarkers, NICRAD leading the way here with centralizing first the capture of DNA, but now also capturing plasma. The development of plasma and PET biomarkers has really changed the way that we can track the disease. And of course, you've heard about SCAN and the ways that MRI is being used in NACC data to track neurodegenerative processes, to track white matter changes that occur, and of course, UDS4 and all of the ways that cognitive data are being measured and tracked longitudinally to look at the ways that Alzheimer's disease and related dementias change over the course of the clinical disease. So my team is really focused on ways that we can use integrative approaches to look for precision medicine markers of Alzheimer's disease. I think a lot of times we talk about this as the right treatment for the right patient at the right time. Often we think about risk stratification when we're talking about precision medicine, but I want to talk a little bit about how the right target is really important here too. And if we think about risk stratification within our analytical models, we can end up with different answers for different populations, and maybe the right treatment really will be different in some of the stratifications that we're all very familiar with in Alzheimer's disease. Yesterday, you heard from Dr. Wang about the different Alzheimer's disease sequencing project programs that have been initiated. I'm very excited to present right after two studies that we're talking about artificial intelligence and machine learning, because one of the things that we have to do a lot of times before going into those models is data harmonization, and it's not a lot of fun. It's what most of my career is focused on, so I'm going to tell you a little bit about what we're doing on the data harmonization side and how NACC has really facilitated that. And then, of course, the functional genomics is the way that we go from a marker to a target. So let me tell you a little bit about the Alzheimer's disease sequencing project phenotype harmonization consortium and how NACC has in particular contributed to this. So the goal of this effort is to take data from more than 50 cohort studies that are in the Alzheimer's disease sequencing project, to curate that data, to document the way that data was collected, to harmonize it, and then to make all of that available in the resource you heard about yesterday, NIGADS, the data sharing service for genomics data at the NIA. And we're also integrating data that's already being captured by NACC and other studies that are in LONI. So we're really not building anything new here. We're using established infrastructure that NIA has invested in over the past few decades and harmonizing data and then making it available right on those resources. NACC has been really important for this for a number of reasons. I'm just going to highlight a couple of them quickly. For the harmonization project, rather than developing new methodology, we went with established methods that had been published previously with domain leaders that you can see here, all of whom have built their careers on establishing really great methods for harmonization. I just want to highlight a couple of the modalities that NACC has been really instrumental in, particularly the PET harmonization, the neuropathology harmonization, and the cognitive harmonization. So we had our first data release in the fall last year. You can see that NACC was a huge contributor to this. These are all participants that have whole genome sequencing data already available on NIGADS. All we're doing is making deeper phenotypes available as well. You can see NACC contributed over 10,000 participants to that initiative. We released harmonized cognitive data, harmonized biomarker data, and harmonized neuropathology data. We're going to release our first neuroimaging data sets this year. And if you are working on AI or machine learning approaches, please reach out to us because we're trying to release some of those curated collections even earlier this year. So if that's a space that you're working in, we'd love to talk to you more about that. There'll be more than 20,000 images that we're releasing in this data release coming up this year. So we'd love to talk more about that. I don't have time to talk about it today. You heard Dr. Mormino talk about the SCAN initiative and the way that they're collecting data in a harmonized fashion through SCAN that's really going to transform what we can do on the genomic side. She didn't have time to talk about the legacy acquisition that they're doing for this project as well. One of the things that's really cool about NACC is there's so much heterogeneity in the way data has been captured that Dr. Mormino's team has been able to test out pipelines that we can now roll out to all these other cohort studies, many of whom use different acquisition protocols as well. We already have a test bed, we've already validated that it works, and now we can start pulling in other data sets. NACC has been really instrumental for that effort. You can ask Dr. Mormino more about that. I probably can't take questions on it. The neuropath harmonization led by Dr. Montine and Dr. Beecham. Again, in this case, we have cohorts that have collected neuropath data in very different ways. We get everything from neuropath forms coming to us with no structured data at all to structured data that's been collected in a very different way. NACC neuropath forms are the gold standard that we map everything else to. So Dr. Beecham and Dr. Montine will go through all of that data, map it back to the NACC forms, and that's what we end up releasing is things that are aligned with NACC. So NACC, of course, has been absolutely instrumental for that. And you can see some of the ways that Dr. Beecham has recoded other data sets to align with NACC. And these data, we released about 6,000 participants this year. That number will increase a lot more this fall, but you can already access those right on NIGADS, and those are all NACC neuropath forms. I'm going to talk very briefly about the cognitive harmonization to make the point that even though we're releasing 15,000 participants on NIGADS, we're actually harmonizing data from the whole cohort. So the cognitive harmonization included 55,000 participants for these first 4 data sets that were released in the fall. And the way that Dr. Crane and Dr. Mez do this is they go painstakingly through every item that's collected in a neuropsychological protocol. They make a decision about what domain it should be in. Then they make a difficult decision about whether or not an item was acquired in the exact same way across studies. If it is, we can use that as an anchor, and we can build these nice latent variable models that give us composite measures of cognition that can be used for longitudinal analysis. In this first data set that we released, I'm just going to show you some of the manuscript for this was published last year, led by Joey Mukherjee and Dr. Crane. Check that out for more on the methods. But just to give you a sense of what these types of data can be used for, even now with just our first 4 data sets, we ran a GWAS, and for those of you who are geneticists, you'll recognize this Manhattan plot. It looks a lot like the Alzheimer's disease Manhattan plot. You can see with just 20,000 participants, we're able to identify a lot of established markers of Alzheimer's disease, suggesting that memory performance, of course, is a great endophenotype for Alzheimer's disease, and it's going to expand our ability to identify new markers. But what I'm really excited about is the ability of, as NACC is increasing diversity, increasing representation in the studies, and we're bringing in more and more representative studies, particular Spanish-speaking studies, we're going to be able to ask intersectional questions about where sex, gender interact with race, ethnicity, for example. So one example, this manuscript was just accepted a week ago. We looked in the first 32,000 participants that we had harmonized data on, and we confirmed, no surprise, that there's a sex difference in the E4 association with memory performance. We also demonstrated for the first time that that extends to the language domain, and importantly, that effect is actually present in both non-Hispanic white and non-Hispanic black participants. And I'm really excited as the diversity increases more that we'll be able to ask that question about other ancestral populations and start thinking about ancestral haplotypes that could be contributing too. Just one example, this is a manuscript that's under review. We looked at the X chromosome for the first time in relation to some of these longitudinal phenotypes and identified an X chromosome locus that was associated with memory performance only in non-Hispanic black women, which again I think is really an exciting thing when we're thinking about target identification. We've got to get sample sizes a lot bigger if we're going to be able to identify a target that's relevant within some of these very important stratifications. I'll just highlight one more thing very quickly. I'm really interested in resistance and resilience to Alzheimer's disease. The way that we do that is we use a residual approach. So we'll have amyloid and tau pathology predicting cognition, look at the residual for people who perform better or worse than expected. We use fancy latent variable models to calculate a resilience phenotype. And what you can see is that people who are high resilience on the x-axis decline slower than people who are low. But most importantly, that's particularly true for people who are biomarker positive. Well, a huge limitation of this approach is I have to have either an autopsy measure of neuropathology or a PET or CSF marker of neuropathology. And of course, most data sets that have that are largely non-Hispanic white. So one of the things we've been working on with this new data set is, can we build a silver model where we use all the data that we have and we don't have biomarkers to try to estimate resilience measures and then confirm it in data sets where we do have pathology. And NACC is a critical piece of infrastructure to be able to do this. We can build a model in 20,000 people, confirm in the 5,000 people who have neuropathology data that our resilience measures are actually working. And when that's the case, now we can go in and do a new GWAS and identify new loci. In this case, we identified a cluster of genes that are really important for autophagy, and we think are very important for resilience in particular. So I know I had to run through that, but hopefully that gets you excited about some of the ways you can use NACC data. Thanks to Dr. Biber for partnering with us, and thank you for listening. Thanks to all of my collaborators, particularly Dr. Archer and Dr. Dumitrescu, assistant professors on my team who have really led a lot of these efforts. So thank you very much.
Wonderful job, wonderful job.
I'd like to call up Dr. Barnes and Dr. Barbino and Dr. Das and Dr. Rabinovich and also Dr. Kolachama and Dr. Rosavian to join me on stage for a Q&A.
So we only have about 4 minutes if we go just 2 minutes over time.
You're going to give us extra couple minutes? Okay, so let's take a few questions.
Hi, I'm Adam Brickman from Columbia University. Congratulations to everyone. I'm blown away by how much work is being done. It's really cool to see. I think my question is for Beth, and maybe it's for NIA program, about how some of these SCAN and Clarity initiatives are going to fit in with the new RFA, about requirements for PET scanning within the ADRCs. Do we use our scans from Clarity to count towards that outcome, or is this in addition, or is there other synergy there? Thanks. Boring question, I know.
I'm going to talk about it later.
I'm Krista Mulder from the Echo Night ADRC in St. Louis. Question for Dr. Rabinovich. I was wondering if you and your team have thought about the consent process for ALZNet versus at the ADRC, given that one could enroll in one versus the other temporally, and how that consent process would work?
Yeah, I think ideally it would be streamlined and you could have everyone do one consent. I think the trick, of course, is that as I mentioned, ALZNet data really needs to be entered by a treating provider, by someone who is prescribing the drug and monitoring safety, and that will happen outside the ADRC. And so this is really part of what we want to hear in the questionnaire that we'll send out, and the webinar is how this might work at different ADRCs and how that interaction and collaboration might work. In our center, there's a very close relationship between ADRC investigators and clinicians. Basically, all the ADRC investigators who are clinically trained are also active in our clinic, and so you can imagine a good interaction and synergy. But I know that that's not the case, that there are other centers where clinic and the ADRC are very separate, and so I think there'll be some fine-tuning and probably different solutions. I'm not sure there'll be a one-size-fits-all solution for that question. It's a great question and something that we're talking to Sarah and others at NACC about actively.
Okay, is that it?
I think so. We don't have any from online. And so I do encourage
There's
you
one more
all-
in the back, Hannah, hold on.
Okay.
I'm Rich O'Brien from Duke UNC ADRC. As far as integrating this into real-world clinical practice, CMS just came out with the new memory code that most people are using, and just 2 weeks ago announced that we have to use scales that they're determining, which don't include the FAQ, the NPI. They're scales I've never heard of. Is anyone aware of what's happening at CMS?
I will say that as part of our COVID-19 supplement work, we are looking at integrating CMS data through MEDRIC, and that is something that is well underway and will hopefully serve as a model for potential ways that we could do that broader moving into the future. But that's, I don't know if others have other things to comment on that, but.
I'm not aware of the specifics, but I think it will be, even in ideas and new ideas, our experience has been that it's been very difficult to get different centers to even agree to do the MMSE or MOCA, which are widely used. And so if there are new instruments that are less familiar to clinicians, I think it'll be a heavy lift to try to convince people to use them.
Okay, one last question.
Sudha Seshandri, Stack, South Texas. Want to congratulate all of you on the many ways you're modernizing NACC. I had a question about the identifiability of voice. Even if you scrub it of information like my name is so-and-so, my voice is my voice. How, when we talk about widespread data sharing, particularly in a related way, if you're giving iPads to people to record at home, there's a potential for those to become portals for hacking. And I'm sure you've considered that, but we have questions locally at our IRB.
Yeah, this is something we're going to be collaborating really closely with the new tech work group to think about concerns like that. We don't currently have any voice data, right, as part of our data repository. It's something we're interested in doing. I'm sure Rhoda Au, Sean Mooney, Andy Sake, and others, I think have thought a lot about this. And, you know, we have a lot of work to do to figure out how to do that right. So we'll be proceeding with caution. Okay, we're done. Thanks, everyone. Sorry for the condensed Q&A.